Hello friends, Ace Botane, next episode. So in this episode, we'll be discussing photosynthesis, first one. Again, a very difficult chapter for most of the students. So for convenience, I will share the screen. Now it will be coming. I think screen came. So this is the first page in which I wrote 3 chapters, of which plant growth and development, respiration in plants, both are over. Now it is photosynthesis in higher plants, among the topic physiology. In this, we can find first page of NCERT of this chapter connected photosynthesis in higher plants, where it generally describes, that is, 2 views, 2 perspectives of, or 2 levels of organization of life forms and phenomena. One is cellular and molecular level of organization. Other is above level of organization. Above level of organization deals with ecology and related aspect, environment related aspect. While the second one, that is cellular and molecular level, it deals with physiology and biochemistry. Now comes that photograph, the famous scientist Melvin Calvin, who received Nobel Prize in 1961 for elucidating famous C3 cycle. That's a part of this chapter. Then on the right-hand side, some description about what the work he done, all this. That we'll be studying later. So I think first page is clear. Now we can proceed to second page. What all things we'll be learning in this episode. 7 topics given. So one by one we can proceed. First topic is meaning of the term photosynthesis. So from there we should be able to understand. Photo means light, synthesis means production. So something is produced by utilizing light. I think that word is clear. Now, second aspect, importance of photosynthesis. This is the only reaction in this world where light energy is utilized. And by utilizing this light energy, some chemical reactions are taking place and oxygen is released. At the same time, food also will be produced. So 2 aims will be satisfied. One, oxygen supplied. Second, food produced. Now, third aspect, oxygenic and anoxygenic photosynthesis. See, when you check our heading, you can find the heading here, photosynthesis in higher plants. Why such a term, higher plants? Because in higher plants, when plants do photosynthesis, oxygen will be released. So it is called oxygenic. But in lower plants like bacteria, oxygen will not be released. So it is called anoxygenic. And remember, in cyanobacteria, it is oxygenic. It is an exception. It is connected with evolution of a popular question. Cyanobacteria, it is oxygenic. Higher plants, it is oxygenic. While bacteria-like organism, it is anoxygenic. I think that part also clear. Now comes chemistry of photosynthesis. Photosynthesis is basically an anabolic endergonic event. It is a physico-chemical biological process. It is an oxidoreduction process where we can find light energy will be finally converted into chemical energy. And the equation has been given for theory question. They will be once in a while asking, write the equation for photosynthesis, 6CO2 plus 12H2O, light and chlorophyll is must, C6H12O, 6H2O plus 6O2. I think that page is clear. Now, when we study photosynthesis, just like respiration, there are certain inhibitors of photosynthesis. Many types are there. NCERT is not directly telling it, but if you know, it is good, 1 or 2 names. Photosynthetic inhibitors mainly includes herbicides connected with the photosystems. Typical example is DCMU, dichlorophenyl dimethylurea, connected with the blocking of ETC, that is between quinone and plastoquinone. And another one is imazine, atrazine, urea derivatives, all this. Now, basic things needed for photosynthesis. 3 items required. One is light, one is chlorophyll, and one is carbon dioxide. And there are certain experiments for establishing all this in NCERT itself, usual theory, practical, neat model questions. So light and chlorophyll, I put it together with a different color. Single experiment will be sufficient to prove light and chlorophyll are needed for photosynthesis. Name of the experiment is variegated leaf experiment. If we cover the leaf with the black paper, not starch test, negative starch test. Suppose no covering for leaves, it will show positive starch test. Now, carbon dioxide we can discuss after this. So what is starch test? Chemistry also will be learning it still. What are the basic procedures? Originally, we have to kill the leaf by putting the leaf in boiling water for 30 seconds. Then remove the chlorophyll by putting the leaf in boiling ethanol. Then soften the leaf by dipping in water. After these 3 stages, keep the leaf on a white tile and add iodine solution. If starch is present, color changes from brown to blue-black in a short time. Blue-black. That depends on concentration of iodine and the medium. It is equally important. This is how the starch test will be performing. Practically, we may be doing it. Now comes next experiment, third one, that is carbon dioxide is essential for photosynthesis. It's a popular question, even practical theory question. Name of the experiment is Mole's half-leaf experiment. Why the name came? Because a well-watered potted plant kept in darkness. Usually photosynthesis experiment be done by keeping the plant in darkness earlier. Then bring into sunlight, attach a leaf through a splitted cord into a bottle. So the bottle contains KOH solution and system is kept. After some time, leaf is plucked and starch test is conducted. We can find the part of the leaf that is seen inside the bottle. There is no starch because KOH is there. KOH has an ability to absorb carbon dioxide. Without carbon dioxide, no photosynthetic reaction. A simple but famous test. Leaf that is seen outside the bottle. There we can find starch. I think idea is clear. So these are some important tests. Now one more that later we'll be studying. Still, oxygen bubble experiment. Purpose is that we'll be learning still to show that oxygen is released during photosynthesis. Hydrilla plants will be taken. Keep the hydrilla plants in water in bright sunlight. Small bubbles around the plant. But in darkness, we cannot find bubbles around the plant. So bubbles only seen when sunlight is there. And the bubbles when tested, it is oxygen. There is a famous experiment, oxygen bubble experiment. Later we'll be learning it still. I think that page is clear. Now comes scientists and their contribution historical account. This is a little bit important. 1 or 2 questions regularly they're asking. Match the following, fill in the blank, theory and runs, NCERT oriented. So we can proceed first with Joseph Priestley. And the jar is, that experiment belongs to Joseph Priestley. 1770 onwards, a series of experiments were conducted. And what he established is essential role of air in growth of green plants. So everything I given there. Essence is plants restore to the air whatever breathing animals and burning candle remove. This type of statement will be given, identify who suggested, comment on it this way. And in this experiment, we use 2 terms, phlogiston and dephlogiston. Phlogiston means not a good gas or a poisonous gas. Dephlogiston, D means minus, remove. It is a good gas or non-poisonous gas. Photograph of Joseph Priestley you can observe, 1733 to 1804. And what he mainly established. One is discovery of oxygen in 1774, chemistry side. Then our side, essential role of air in the growth of green plants. What he observed, that is the experimental part. Priestley observed that a candle burning in a closed space, example is a bell jar, soon get extinguished. Figures you can find it. Similarly, a mouse kept in that bell jar, that will be suffocating and later it may even die. But he concluded that a burning candle or an animal that breathes air, both sometimes damage the air. But when he placed a plant refers to mint plant, in the same bell jar, he found that mouse stayed live and candle continued to burn. That means oxygen supply is there and carbon dioxide will be absorbed. So the last statement, plants restore to the air whatever breathing animals burning candle remove. mass of pollen type question, direct question, theory question, all these historical events. I think that page is clear, theoretical only. Now we can proceed to next scientist, Jan Ingenhorst. What he done is, like previously, sunlight is essential to the plant process that somehow purifies the air, followed by burning candles or breathing animals. So sunlight. An elegant experiment in aquatic plants in bright sunlight, small bubbles were formed around the green plants. Already we done that experiment, oxygen bubble experiment. While in dark, they did not came out. Oxygen did not came out. Later, identified these bubbles as oxygen and only green parts of the plants that could release oxygen. Simple experiment. In aquariums, we can find this bubbles. Scientist is Jan Ingenhorst, 1730 to 1799. Next scientist is Julius von Zach, a regular question. About 1854, around it, Julius von Zach provided evidences for production of glucose. So Julius von Zach, glucose, when plants grow. Glucose is usually stored as starch. Further studies showed that the green substances in plants is located in special bodies within plant cell. And these green parts in plants where glucose is produced, it is usually stored as starch. So green substance and the special bodies. Green substance is nothing but chlorophyll. Special bodies, now it is called as chloroplast. Julius von Zach, glucose. I think that idea is clear. Now we can proceed to next scientist, T.W. Engelmann. So what he done? Few experiments in a green algae called cladophora, connected with aerobic bacteria. So experiment part is, with the help of a prism, he split light into its spectral components and then eliminated a green algae cladophora, placed in a suspension of aerobic bacteria, and he was able to identify bacteria were used to detect the sites of oxygen evolution. And he observed that the bacteria accumulated mainly in the region of blue and red. And these two areas are maximum photosynthesis taking place in daytime. So blue and red light of split spectrum, maximum accumulation of bacteria. And he created first action spectrum of photosynthesis and explained. Scientist is T.W. Engelmann, 1843 to 1909. Then at that time, there was an empirical equation, CO2 plus H2O, light, CH2O plus O2, empirical equation. Now comes next scientist, Cornelius van Niel. He done a milestone contribution. Profession-wise, he was a microbiologist. Organism used is purple and green bacteria. Photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. That is the work of Van Niel. Equation is given, 2H2A plus CO2, light is needed, 2A plus H2O plus H2. And in green plants, H2O is the hydrogen donor and is oxidized to oxygen. Some organisms do not release oxygen during photosynthesis. When H2S is used, as in the above case, oxidation product is sulfur or sulfate. So depending on the organism and not oxygen, it indicates oxygen in green plants comes from H2O and not from carbon dioxide. Further proved by radioisotopic techniques, because that time there was a controversy where oxygen comes from water or carbon dioxide. So Van Niel was able to solve this issue up to certain limit, where oxygen comes from water. I think that page is clear. Now comes to give a concrete proof that oxygen comes from water. That work is done by Rubin and Kamen. They used heavy but non-radioactive stable isotope of oxygen, namely O18 in H2O. So H2O they provided O18. Well, in carbon dioxide, O16. Then photosynthesis was done. They checked which oxygen is coming out. They were able to find oxygen in photosynthesis coming from O18. O18, that experiment is given to H2O. So oxygen comes from water and not from carbon dioxide. Rubin and Kamen. Rubin is 1913 to 1943. Kamen is 1913 to 2002. And there comes the correct equation for photosynthesis. Regular question, theory entrance. 6CO2 plus 12H2O, light and chlorophyll, 6H12O6 plus 6H2O plus 6O2. Still, many workers are there in this field. Mainly, we connected with NCERT. Few more will be there that will be coming in discussion part. I think idea is clear. Now, 5 theory question, and there is 5 NEET model question with the key. So we can just pass through once again. So I will change the screen for you. So we started from here, NCERT, 2 perspectives of biology. One connected with environment, other is connected with the internal studies. Then Melvin Calvin, Nobel Prize, 1961, famous C3 cycle, what all work he done. Then what all things we'll be learning in this chapter, in this episode, 7 items we'll be learning in this episode of the chapter. Meaning of the term photosynthesis, importance of photosynthesis, oxygenic and non-oxygenic photosynthesis, then chemistry of photosynthesis. After this, inhibitors of photosynthesis, then basic things needed, light, chlorophyll, carbon dioxide. For light and chlorophyll, variegated leaf experiment. For carbon dioxide, moles half-leaf experiment. Then we discuss starch test, how it works, etc. Then experimental part, historical event starts with the Priestley's experiment, followed by Jan Ingenhorst. Then Julius von Sack, glucose. Then spectrum connected with Engelmann. Then comes Van Niel, connected with the oxygen comes from water, not from carbon dioxide. Initial steps. Then finally, a concrete evidence given by Rubin and Kamen by using O18 in water, O16 in carbon dioxide, and which O is coming after photosynthesis, it is O18. O18 is incorporated in water. Then an equation. After that, 5 theory question, 5 NEET model question with the key. That is regarding today's episode connected with the photosynthesis, first one. Next episode, I will be doing genetics. Again, after that, photosynthesis alternative, it will be coming. So it will be Wednesday, genetics, then Friday, photosynthesis. Because both year students are asking simultaneously. So this is the only remedy what I can provide, that is why. Okay, so we can meet later. Thank you. If you like it, subscribe or like it, share it, comment on it. So we can meet later. Thanks.